am honored to be invited to give the talk for this event, and especially honored to have members of the Hansen family here. So my topic, let me point out, adaptive sampling. I want to go through a range of spatial and network settings and examples where it's used, and then I have at the end of the talk I have a few short video clips to show, to give some, I think, to give maybe a better understanding or insight into the particularly the changing part, the dynamic sampling and changing populations. That's been an increasing interest of mine for a number of situations. So sampling can be used for a number of methods, and what most of us have worked most on is selecting a sample from a population in order to make observations on the sample in order to estimate characteristics of the population. But in addition, sampling has a role underlying experimental designs. Where do we get our experimental units and the whole thing fitting together, and then in intervention designs more broadly, where we're not just trying to infer the effect of treatments as we are in experiments, but we're trying to affect the wider population. Now, incidentally, in the course of this work recently, I realized that the first sampling problem I worked on was actually, it was in a wildlife problem. There were study methods for bears and other large mammals, how to do that and estimate their abundance without disturbing the animals. At the time, the research methods were to track the animals, stab them or shoot them with a hypodermic tranquilizer and take various physiological measurements and stuff while the animal was aware of it but couldn't move its muscles, and there was some concern that would affect both their health and their behavior. So I realized what I was actually doing was sampling. What I was working on was sampling methods to avoid making interventions with sampling. So today I'm more focused on sampling methods where we want to make the interventions. But a big part of it is the other. So if you think you work in a part of survey sampling where you don't make interventions, you might think again. I think that the most common intervention made in survey sampling, for example, telephone survey and so on, is to change the person surveyed so they're less likely to respond on the next survey, and that's been a widespread problem, and this problem of declining response rates, and part of that is due to just the saturation of, this took longer than they said it would take you to do, Phil. So just that effort, which seems mild, but is actually having leading to a lot of questions about how to do surveys in the future. So sampling for inference. We select a sample of units from the population and observe sample on the nodes. And so our ideal here is to make no intervention at all, to not influence, to not change those values by are amazing. And in an experiment, in contrast, we have a sample of experimental units. We assign treatments to units according to an experimental design. And so we want to make an intervention to change and then to follow through and be able to make inference on the effect of that intervention. And most experiments are done, most experimental designs incorporate the purpose of trying to have the treatment to one unit not to affect other units, and to have and to have the response of one unit not affected by the configuration of other units. And a lot of that is done by design rather than just kind of assumption and hope. Now, in intervention designs more generally, we're interested in making an intervention in a population, like health intervention or integrative pest management techniques in agriculture. We would like this intervention to benefit the population, and usually we can only make the intervention on a sample of the population, on some subset. But even so, we would like it to benefit the population to have an effect that goes beyond the sample. So an example would be where the epidemiologists seek to have some portion of the population, some sample vaccinated, and then that protects other people in the sample from the contagious disease as well as themselves because of the network contagion. So it's quite different. And then the other thing I'm finding, and I'll show some of these examples, is that it's actually kind of a rich study because often there's more than one agent with a intervention design in the population, and the designs or the intervention strategies can interact one with another, one enhancing the other, one suppressing the other, one adapting to the other. So adaptive sampling, the idea here is that the procedure for selecting the sample may depend on the values observed during the survey, which weren't known before the survey, and then the sample itself adapts to what's encountered. So a conventional design, such as simple random sampling or stratified sampling, we think of the design, we relate the design to the procedure for selecting the sample to a probability associated with each possible sample. And in a conventional design, that doesn't depend on other values unless they're auxiliary data known for the whole population before the survey. And in the adaptive design, then the probability of selecting the sample can depend on variable values associated with units in the population, and it might depend on some design parameters or even population parameters as well. So, and then in this type of work, so just as in the 1940s, and it wasn't so obvious to people that unequal probability designs could be used and come up with unbiased estimates. It was actually a bit controversial when I think Morris Hansen and others were pioneering that work. Same with the adaptive sampling, the same problem that if you select your sample in this clever way, then maybe you've biased it in some ways. And so it's again takes an effort to get unbiased estimates or estimates with other desirable properties out of it. And so in my work, in order to do that, I started using the design-based approach, and I increasingly I use the model-based approach, and the two in combination with just a variety of methods that work. So here's an example of the type of thing that was originally motivating these adaptive designs. It was work in sampling for commercial fisheries, fish and animal species and rare species of plants. And the problem with the survey is that if people selected a random sample like this, they found very few values of what they were looking for, you might say. That because of the clustering, the uneven distributions, most units might have, say, a 0 value, and then there's high abundance somewhere. And so the idea was to follow that through, and where we find high abundance, we'll add neighboring units to the sample. We found more high abundance with that, so we add more. So this type of example motivated it. Now you have an example where the sample mean of the sample wouldn't be unbiased, so that you can unbias it in certain ways, and the two ways I used to were related to the Hansen estimator and the Thompson estimator, but the problem here was that we could not from the sample calculate the selection probability or the sample inclusion probability for every unit. So we do something kind of like that, where we're dividing by a network intersection probability, and then I thought this could be improved by the route lateral, but in the late 1980s when I was doing this, I didn't know how to do that computationally. It was too computational intensive. So in the model-based approach, there's some clarifying work by Don Rubin and some others that I think laid it out really well toward the end of the 1970s for both sampling and experimental design on how the sampling design integrates into the likelihood function, so it affects like maximum likelihood estimates or Bayes estimates, and only under certain conditions does the sampling design part factor out of the likelihood. In the controlled adaptive designs I was working on, it does, but in the more complex cases, it doesn't. So there's kind of a parallel effort in estimation, inference and modeling going together using these ideas. But the key result to me that jumps out of all this is that if we have some data to analyze, you really need to know something about the design in order to be on a firm base in doing that. And where the design turns out to be ignorable at the inference stage, most often that's because people have paid a lot of attention to the design and done it specifically so that would be so. So I work a lot on these spatial and ecology and environment type examples motivated a lot of this work. And starting in the mid 1990s, I was people asked me from NIH and from CDC if some of these same ideas might work with the hidden human populations. They were thinking of the people at high risk for the spread of HIV/AIDS, for example, and and it's very much related, and the secondary sampling in networks, and so with a network we have a set of nodes and a set of links between them. So we can think of the nodes being people and the links being social relationships, and often close relationships on which the disease might spread. And so we have, in addition to units, the usual units in the population, and the variables of interest associated with each unit, we also have links in the population, and also variables associated with them. So presence or absence of a relationship between people, and also what type of relationship, how strong, things like that. And the problem now is that we're following, the only way they can find these hidden human populations often, the only way they can find many people, they can find a few to start, and then they follow social links and build a sample that way. And then they who have you been doing this activity with, or who do you spend time with, and build their sample that way. And so our population now has a network structure, and our sample is a sample from that network, and it's selected in kind of a funny way, which is actually in most cases adaptive. And this is done for practical reasons that people find it hard to understand. So this also has design-based and model-based approach, and we'll just say that here in the design-based approach, the values associated with the nodes, the set of nodes, and the values associated with the links and where the presence or absence of all those things are considered fixed, and probabilities coming just from our design running through there. And with the model-based approach, the appropriate kind of model is a stochastic graph model or stochastic network model. Let me just, let me just speed up here. But so here's one type of network sampling design that I worked on a few years ago that has the form of like at each time step, our sample has some number of links out from it, and we'll select one of those at random, or one of those by probability sampling, and then at the next time step, we'll select another, and so on, and so that we don't get stuck in a single component of the network that's not connected with others. We'll leave some small possibility for a random jump at any time too. And so here's an example. If this is our population, and I take a node or a unit at random, and it has no links out, so I take a jump. And this one had links out, and I just start kind of in a free way. I just keep building my sample. This is kind of fairly intuitive. It's still not quite the most natural thing to do in the world, because we're just taking one jump at a time, and often it goes much more freeform than that. But that's a type I worked on, and it had an estimation challenge. So now that I've done this, these might be like a population of people with a high risk, some of whom have a high risk behavior, such as injecting illegal drugs, on which the HIV transmits on those links in some places. And so again, there was a design-based and model-based estimator. It took a different type of estimator this time. I couldn't use the same ones because of the probability structure, but the first approach I followed was the design-based one, which just said, find some simple initial estimator. Like we selected the first node of the first set of, the first node of the first set of nodes at random, so the sample mean, say, would be an unbiased estimator of the population mean for that activity. And then we can improve that using the Rao-Blackwell method, where we take our initial simple estimator and calculate its expected value given our sample, given the minimal sufficient statistic for our sample. And what that meant was we had to figure out all the different sample paths by which we might have got the same sample. So it's a simple idea, but for medium-sized sample sizes, very soon there's a very large number of these paths, and it's a computational problem. And so, so let me go light on some of the details of these estimators, but just say, in the process of this work, I found a practical way, at least up to medium sample sizes, of calculating these Rao-Blackwell methods. And that was using a Markov chain Monte Carlo technique where I would resample from the data as if the data were the whole population, and I would apply the same sampling design, the same link tracing network sampling design to the data, and then start averaging that over the different sample paths. And it takes an MC check to, you might say, adjust to the fact that the probabilities are different within our sample than they would be selecting that same unit from the whole wider population. So, and here's the other thing I ran across doing the network. Once I solved that problem in the network sampling, I found I could recast my spatial sampling problems as a network sampling problem in the types of cases. So here's my, rare clustered animals or whatever, and if in the course of sampling we find an area with a unit with high values, we'll add neighboring units. That corresponds to a graph structure, a network structure that looks like this. It's a directed graph with where in some cases, the node in the middle there, say, would lead us through the arrow to the node to the right of it, but the node to the right would lead us back to the node to the left also. And in other cases, it's a one-way route where this sampling this unit would lead us to then look at the unit next door, but the unit next door, because it doesn't have an interesting value, would not lead us back if we sampled it first. So it has this structure, and so I'm able to apply the same type of network design in it by following those links. I started with a random sample of nodes, which are actually the sample of those square geographic units, and I had neighboring things like this, and I ended up with a spatial sampling. And that meant that the same estimators applied, which added some flexibility. So in the network, and so those were the simple design-based estimates. I would say that the model-based estimates have maybe been the most successful. They're the ones that are spreading because the network sampling situations, many of them are just too complex to find good, efficient design-based estimators. The design-based estimators put some constraints on the kind of sampling you can do and even how complex the model is. And so the Bayes estimator, where we use Markov chain Monte Carlo techniques to sample from the predictive posterior given our data, have been the most successful, and people are, I think, a number of researchers are advancing that work anyway. But the basic idea here is I have here I'm using theta for node parameters, like what's the probability somebody injects drugs, and I'm using beta for for link parameters, like what's the probability of a link between these two types of people or these two groups. And the idea here is we go through MC with a data augmentation step, imputing the whole network from the data at that point, do that a few thousand times, and it's just very flexible as far as what the type of inference we can do from that. And so here's an empirical example I worked on with some of these, and that's it was a study out of the health department in Colorado Springs, funded by the CDC in the early 1990s on the heterosexual transmission of HIV/AIDS. And so it was actually sexual and drug use transmission in that area. And but they, for my purposes, and it was good, because they collected a lot of data on a somewhat complete network. And I'm using the core of their data here, and it's drawn to bring out the clustering. It's laid out visually. It's laid out for visual meaning, if you want to put it that way. But here's what the data look like. The red nodes have the high risk behavior. I think it's the injecting drug use in this case. There's the links between people, and we see there's a really large component of people who are all connected through this. So potential for an epidemic explosion that hasn't really happened. So I could use that to simulate. I could select a sample like this through the network sampling designs I was showing, and then estimate different things like the degree distribution, or the various things about the rates of high-risk sexual behavior and things like that. And so if you take the sample data at face value and say, well, I followed all these links, and 50% of these people inject drugs, but it might be that 22% of the people inject drugs, and you have to, but your sample has enriched itself with the more high-risky behavior, because those are the people with more of those kind of links, and so there's a higher probability that they come into the sample. And for the last few years, a lot of my attention has gone into this and on the interventions in sampling. So think of something like the integrated pest management was actually one of the first cases this came to my attention. But then I was working in this HIV/AIDS, and the purpose of all this attention on the survey methods for the hidden human populations and the network sampling and everything, the idea is then we would understand better the behaviors and the geographic distribution of those behaviors and where interventions were needed, and kind of separating the two. And I started kind of I started to imagine putting that together with what if your sampling program in the first place is focused on making interventions, and what might we do? And also these populations all change. They go back to the same people, but the network components have broken apart, and some people have moved away and others have died, and the study population is almost changing as the study goes on. So now we need a sampling process. We have to conceive of the population as like a stochastic process that changes through time. If it's a network population, relationships form and relationships dissolve again. People come into the population and people move out of the population. They're born, they die, they grow up and come of age that the study is considering. They move in from another area, they move out, and so we have a stochastic population, and then the sample is itself a stochastic process. We have like a random set, if you like, which is our sample. There's a selection process that is an acquisition process by which nodes are selected into the sample, and a detrition process by which nodes are selected out of the sample. So everything is changing, and we want to make an intervention by selecting a sample of nodes and applying intervention to those samples. So in the HIV epidemic example, the type of network designs we're thinking of. First of all, there's the seek and treat designs where they test people and see if they need. They follow links and they follow links from people they know have been infected, and they try to get their partners tested, and if they test positive, then they apply antiretroviral, and if they test negative, then that's where we would like to have a vaccine. So we've got some active area of research, a lot of approaches to getting vaccines. I worked with those people for a while on this, but the virus itself has a design that's uncannily similar to the ones we're talking about, where it selects a sample of people. It does it by following links, in this case the sexual links and the drug-using links, and so it uses a network sampling design very much like the same kind I'm looking at to intervene against it, and it applies an intervention which is infection that changes the person, increases their mortality rate, for example. That changes the network. Over time, you see those changes, and it adapts to interventions made against it. That's why there's no vaccine yet. They've had vaccines that look successful, but the virus quickly makes resistant forms, and the same with the retrovirals, and I'll show how I'd say it adapts to the temporal network geometry. Just so everybody knows how I think that is. So here's the stochastic process representing the population. It's a stochastic graph that changes in time, and so the actual set of units changes, and the values associated with those units change, and the set of links between units change, links or edges, and values or variables associated with those links change. Everything changes in time. And here's the sampling process, which at any time t, and our sample is a subset of the population process. And generally we have to think now about how does the sample tomorrow form from the sample today? Some units are added, some units are dropped, some units die or deleted from the population. They disappear from the sample, and so all these things work this way. And so where does the network come from in the first place? It's from people sampling. It's from one person making decisions about forming a link or not forming a link. And so we conceive of that in a social space. That's one way of modeling networks, and where people are more apt to form a partnership with people who are close to themselves in social space. Similar, of course, if it's heterosexual partnerships, they're dissimilar in sex and gender, and then it can also depend on values associated with the nodes and the links, and so the type of designs I look at in this section, a real simple example would be one where I have my current set of units that are in my sample. Those units have some links within themselves, and they have some links going out to units that aren't in the sample. And so at time t, say, we're gonna by some means select a sample of those units going out. The simplest is just it's a Bernoulli sample. They're independent. It might depend. They might have different probabilities depending on values of the nodes and the links and so on. And then links go out of the sample through. They can be removed from the sample, and like if we have an ongoing survey population, like in a panel survey, for example, we might just say, okay, this person's been in long enough, we'll remove them from the sample. Or it might be, that, you know, if the person dies or moves away, they're deleted from the population. They're automatically gone from the sample. So with HIV selecting its sample, because there's currently no cure, no practical cure for HIV, we conceive of that as a without, we say the only way it can be deleted, the only way it can be removed from, the only way it can be selected out of the sample is through the death or moving away of the person. But that will be changed when there's a cure. So here's an example of the type of thing, and then I'll show. I'll run through this, and then I'll just quickly show these video clips that I think make it more clear. So at any point in time, we have a sample, and if you see here, I'll go back one step. We have this sample, and you'll see some units are added to the sample, the red units in the sample, some are deleted, and the sample size goes up and down, but stays about the same, stays in a stable distribution. So let me, so I want to thank the NSERC of Canada and CDC in the US and NSF and NIH for support for this work. And I'll go back, I put a couple of references on that. A background paper in survey methodology in 2011, and then a working paper I posted, and I'm just in the process of posting a revised and second revision of that. And that's just on an archive for now. So we can have the clips of the sampling and its dynamic. Could that be made larger? So here's the one I was just clicking through by hand, but it keeps on going. So it's a stable process, like has evident stationary properties. Let's say the sample size. Here's in the light blue at the bottom. That's the sample size over time. You see it goes up and down, but it stays pretty good. So this is just an idealized design that we do. We follow links by opportunity, you know, with Bernoulli probabilities, and we remove. And so that adds nodes to the sample, and then we adaptively remove links with an adjusting probability just by Bernoulli probabilities being removed, and it comes in balance. And, yeah, we can just go on to these one after the other. So I think that F11 or something. Here's a larger network of this would have about 1,000 nodes, and I'm running one of these fast mixing design. I'm using these designs to measure things about the network. It's the simplest form of design-based inference. In a sense, the design is just measuring its own properties. Now, in this network, you notice this design is spending most of its time up there. What am I doing now? I'm averaging recent values of the design, like a moving average. I actually use an exponentially weighted moving average smoother, and so a unit is either in or out of the sample. That's 1 or 0, but those get averaged, and So if the network was stationary, if the population was static, these probabilities these averages when we kept this going would measure the inclusion probability for that unit in that network. But the network is changing. Those, the number of links a unit has changes and things like that. So this kind of tracks that, and it points to areas that might explode. And when you could go back to that one and just click in the middle so that we see the end part. Down at the bottom, you can click in that bar, and it should. You see, it's spending most of its time up there. That's a highly connected part of the network. It's a certain type of explosive connectivity that that we're trying to measure. And so, with the idea that that's the area where if you light a match, if you get one infected with HIV in there, that's where it can explode. And that's what happens when you see the black dots, as you can see in the middle of the node. That's an infection, and in that area, right near the end, it explodes. But I'll show a clearer view, a longitudinal view of how that time. That's what I'm trying to anticipate with. So these designs, through their average, in one way they're just measuring something about themselves, their selection probability. Another way, they're telling us something about the network, because the design preferentially finds its way more often into these parts of the network. So this is the next one. And so here's exactly the same situation, but this time at the longitudinal one. That could just be minimized. Yeah. So just let that run. That's fine. Now we're looking at the same thing from the side, and we're only looking at the nodes that have the infection. So think of drawing a node on this wall on the left at time 0, and we draw 1, 2, 3 nodes, and now there's 4 in the epidemic, and time is progressing in this direction. And so and therefore it explodes. Now, here's why it explodes. You can see it clearly here. HIV has evolved a high early rate of transmission in the first stage. Also, this is the stage before they're able to test a person for it. It has a low rate of transmission most of the time, like less than 1 in 1,000 per event, like per sexual contact. But the red indicates that early stage, when the node is in the early stage, and it's at the and it's there. That's what gives it the explosiveness, and that's what I'm trying to measure with those fast-moving, fast-mixing designs that get like a moving average kind of that type of place, because once it hits an area like that, it spreads by this higher early rate, and while it's still in early rate, it spreads to 2 more, and it just explodes like that. And then you get back to a slightly more stable situation. So this is how what I call HIV adapts to the network geometry. If I don't fix that early rate, but I just let it select itself, a strain that can do this out-competes the other strains, and it ends up looking like this. So, and then next. So if I run out of time, I can cut these off, but I think there's about 3 more. Here's in contrast. Here's 20 random walks through a dynamic network, and they also, and here I'm averaging them. They also form a measure of importance, like centrality in network. But notice it's not as highly centered as some of those others work with. It's more spread out. So that's the random walk for the same thing. We can just go on to the next one if we're short on time. And here's the components of the network. Notice the components change over time. This is the same network through time. The largest component is red. But the explosion, remember, was in the next largest, the blue one, and that's what this fast mixing network sampling design was anticipating. And that's what the virus, which people think of as spreading real slowly, that's how it can have, you might say, it has explosions in slow motion, and they tend to, they tend to be already exploded before people really catch on that it's happening in a new area. But here's the components. That's not telling us the same. The components are all connected, and so changes and stuff. But that's not telling us what's happening within the component. So it's not the largest component. It's some other component where the real action is here, and then there's one or two more. Yeah, and so those were like 2.5 years, but the 100-year run of an epidemic in an area like that looks like these are the type of things that looks like that. It must repeat, but that's the same. The advance of the epidemic, and you notice very often they tend to go into a kind of equilibrium distribution, and there's things that keep it from going higher, things that keep it from going lower. That's without any intervention to bring it down. And then I think there's one more, because the problem with the interventions that rely on the testing is that the testing misses the early. I mean that you say the testing. So here's one, and the HIV has a clever strategy by having an early stage. There's a cost to having a high virulence for a virus. It increases the mortality of the host. That decreases these connections for it in the network, and it slows the spread. But here's how we try to counteract what would be its advantage. There's people have suggested, an epidemiologist from San Francisco told me people were talking about this, and I found some of the health departments, they tell people in the high-risk populations, in the very high-risk sexual populations, that well, if you would use it, they also use a condom, right? But if you use a condom in just the first few weeks of the relationship, 12 weeks of a new relationship, and then both partners are tested, and then if they test different, they'll keep on using the condom. But if they're both negative or they're both positive, they'll stop using the condom. And that's this strategy here. So you see, there's a lot of relationships. The line between, this is not, what I did here is it's not just condoms, it's any kind of safer sex. That, there's a whole variety of methods as you go to the literature. And if they do that for the first 12 weeks, that reduces to about 10% the transmission rate. And you can see that with the cross-line. So there's a lot of people here not using the safer sex. They're only required to use it for a little while, so it completely stops the same explosion. You can kind of see how that has a. So again, we're trying to take advantage of that same network geometry that leads to the explosiveness and counter it with the minimum effort or the minimum inconvenience. So I think I've used up my time. But that gives I think a picture of the possibilities of the range of these designs and why it brings us into networks and into things that change and move over time in the sample.